So, a gene is transcribed, it's processed, it leaves the nucleus, ends up in the cytoplasm, it's translated, it creates a polypeptide chain of amino acids that folds locally into secondary structures and globally into three-dimensional tertiary structures, and possibly interacts with other proteins through non-covalent and covalent bonds to make quaternary structures. It's going to work, right? Not necessarily. Just like we regulate when transcription occurs, when and how RNA is processed, when and how it leaves the nucleus, and when and how it's translated, we also regulate whether or not a protein will be active. There are general mechanisms through which protein activity is regulated. The first of those is through proteolytic cleavage. And I don't mean degrading proteins, that's going to come later. I'm talking about a protein is made in an inactive form and it requires some sort of proteolytic cleavage in order to activate that protein. The inactive protein, in this case, is turned on through proteolytic cleavage. A second general mechanism is through binding of some other molecule. Molecular binding can either activate or turn on an inactive protein, or it could also potentially inactivate an active protein or turn it off. That same is true for the third general mechanism of protein function regulation, and that is through the covalent modification of proteins, covalent addition or subtraction of phosphate groups. This is known as phosphorylation. Phosphorylation can be responsible for turning an inactive protein on or for turning an active protein off. Oppose this to binding of GTP. There is a special class of proteins whose activity is regulated by GTP binding. In the case of this family of proteins, binding of GTP always turns on the protein. And finally, it is possible to regulate protein activity by simply degrading a protein. This occurs when we know for certain that we are no longer going to need a protein. The other mechanisms are typically used when we want a protein to be at the ready so that we can turn it on and or off quickly. Talking about the first general mechanism, proteolytic cleavage, we see an example on the left of our screen. Zymogens are proteins, or proteases specifically, that are going to be activated by a proteolytic event. Chymotrypsinogen is a digestive enzyme in the small intestine, and it is released by the pancreas to the small intestine in an inactive form, chymotrypsinogen. When we want to digest things we've eaten, trypsin is also released. Trypsin is a protease enzyme that will cleave chymotrypsinogen to produce chymotrypsin, the active form of the protein. Another example of a protein that is activated by proteolytic cleavage is insulin. Insulin is secreted as something called proinsulin, a single polypeptide chain with disulfide bonds holding it in that sort of loop structure. A proteolytic cleavage will remove part of the protein, shown or labeled as the C chain. Two proteolytic events, one at either side of the C chain, will liberate the C chain, leaving behind the disulfide bonded insulin protein. Blood clotting is a physiologic process that depends upon the activation of inactive proteins through proteolytic cleavage. We'll look specifically at factor VII. Factor VII is one of the proteins in the blood clotting cascade. Factor VII is created as an immature protein that requires two proteolytic events for activation. The first removes what's called a leader sequence, and that processing event occurs inside of the cell. Once the pre-pro leader sequence has been removed, an inactive, though mature, factor VII is secreted into the blood. It's not active, however, because we don't want to initiate the clotting cascade when we don't have an injury. However, when we do, we want factor VII to become activated so it can activate downstream clotting proteins. In that case, cleavage of factor VIIa into factor VII occurs in the blood. This cleavage activates factor VII so it can go on and activate downstream enzymes. The second general mechanism of protein activity regulation is through molecular binding. A protein is folded into its three-dimensional shape, and in that three-dimensional shape, there may be a binding site or a pocket into which some sort of molecule is going to bind to regulate the activity of this protein. This brown blob binding into the binding site is highlighted in the larger image, and you see that the molecule that is binding is labeled cyclic AMP. Cyclic AMP forms non-covalent bonds with the amino acid side chains that happen to be sticking out into this binding site or binding pocket, and that binding regulates the activity of this particular protein. The molecule that binds into the binding site is known as the ligand. And again, the ligand is forming non-covalent bonds with amino acids that happen to be sticking out into the binding site. I want to highlight that this molecular binding depends upon random events, but those random events, all the stars have to align. We're going to look at molecule A, and in the crowded environment of a cell, molecule A can randomly bump into molecule B, molecule C, or molecule D. In each case, some number of non-covalent bonds are going to form. However, in the cases of molecules B and C, there are not a sufficient number of non-covalent bonds that are going to hold the two molecules together, the two proteins together, for any great length of time. So those complexes will rapidly fall apart. However, when molecule A encounters molecule D, a larger number of non-covalent interactions are going to form. They therefore will hold molecule A to molecule D for a longer period of time. Your book says that these molecules will stay bound together. However, these molecules will not stay bound together forever. Non-covalent bonds are not glue. These non-covalent bonds can unform just as rapidly as they form. Therefore, when we talk about molecular binding in biochemistry class, we talk about on rates and off rates that will determine how likely two molecules are going to be found bound together. I also want to highlight that molecular binding can both activate and inactivate a protein. Here we see a green jaw-like protein, and that green jaw-like protein can bind to glucose in its active site. So it's going to do something to glucose, but it's also going to bind to another molecule, in this case ADP, at a non-active site that we're going to term an allosteric site. An allosteric site is simply a binding site that is not the active site. We call this allosteric binding. In the left-hand sort of rectangle, we see that of all of those green jaw-like proteins, a small number, 1, is actually bound to glucose. And if it binds to glucose in its active site, it can act on glucose at the active site. However, the vast majority of jaw proteins in this rectangle are not able to bind glucose. However, in the right triangle, in the presence of ADP binding to that allosteric site, now all of the jaw proteins are bound to glucose and acting on glucose. ADP positively regulates or activates green jaw protein by allosteric binding. Conversely, this jaw protein is going to act on glucose binding in its active site, but rather than ADP, will bind molecule X at a different allosteric site. In the left-hand rectangle, in the absence of molecule X, we see that all of the proteins are active, binding to glucose and acting on glucose. When molecule X allosterically binds, it negatively regulates green jaw protein, such that now green jaw protein cannot bind to glucose and act on it. I do want to highlight that there is one green jaw protein that is bound to glucose, and this green jaw protein is not bound to molecule X. There is no way that green jaw protein can be active or acting on glucose if molecule X is negatively regulating its activity. The third mechanism by which protein activity is regulated is through phosphorylation. We talked last time about how amino acid side chains can be covalently modified. Specifically shown here is a serine side chain and the terminal hydroxyl group. What are the other side chains that terminate in a hydroxyl group? Any of those 3 amino acids might be covalently modified by protein kinase enzymes. Protein kinase enzymes are responsible for transferring a phosphate from ATP to OH groups on amino acid side chains in proteins. This is the process of phosphorylation. I'll say it again. Phosphorylation is carried out by protein kinases. This is not an irreversible modification, however, because a second class of enzymes, protein phosphatases, are responsible for removing the phosphate from an amino acid side chain, leaving the amino acid side chain with a terminal hydroxyl group. Notice that there is no information on this slide showing you whether or not the phosphorylated protein on the right is active or inactive, because either can occur. In the top figure, you see the protein, the green sphere protein, is off in the absence of being phosphorylated. However, when a protein kinase acts upon it and transfers a phosphate group from ATP, this particular protein is turned on. Removal of that phosphate group by a phosphatase enzyme will revert that active protein to its inactive state. Conversely, on the bottom part of the figure, we see that the non-phosphorylated protein on the left is on or active. In the presence of a protein kinase, a phosphate group will be transferred, and transfer of that phosphate group converts the active protein to its inactive form. Activity by the phosphatase will remove the phosphate group, and removal of that phosphate group reverts this protein to its active form. The reason this works this way is transfer of a phosphate group results in transfer of a negatively charged group. Addition of a negatively charged phosphate group to an amino acid that is not charged ultimately will interfere with that amino acid's ability to interact with its normal binding partners. That interference changes the three-dimensional shape of a protein, and that shape change affects activity change. The fourth general mechanism of regulating protein activity is through binding of GTP. I told you that binding of GTP is by a special class of proteins that we're going to call G proteins. G proteins not only bind GTP, but they also have the ability to hydrolyze the GTP that they have bound into GDP. Walking through this process, the GTP-bound G protein is active on, and we know that because your textbook uses little red glowy lines to indicate that activity. Hydrolysis of the GTP to GDP turns the protein off, inactivates the protein. So in this way, the G protein is able to turn itself off by hydrolyzing the GTP that it has bound. Eventually, the GDP that it has still bound to it will be released, although that process is slow. But because the concentration of GTP is higher in the cytoplasm than the concentration of GDP, once GDP is let go, a GTP can hop back into the binding site, and that GTP will turn the G protein back on. Unfortunately, the activity of G proteins is not this simple. That is because there are other classes of proteins that act upon G proteins. One protein, a GTPase activating protein, increases the rate of GTP hydrolysis, and by doing that, it inactivates the G protein faster. A second class of proteins affects the rate of letting go of GDP and the subsequent grabbing back on to a GTP. This class of proteins are known as guanine nucleotide exchange factors. Guanine nucleotide exchange factors expedite the release of GDP and thereby the binding of GTP, and that activates G proteins at a faster rate. Finally, I do want to talk about protein degradation as a means through which we can get rid of proteins when we know that we no longer need them. I'm talking about proteins in the cytoplasm, not proteins that have been ingested through endocytosis. If a cell has made a protein that it wants to get rid of, it no longer needs, it will use the non-membrane bound organelle, the proteasome. In this central yellow highlighted core of the proteasome are a large number of proteases that will digest proteins into their component amino acids so they can be recycled. How does the proteasome know which proteins the cell no longer needs? We tag them. Our cells will tag proteins targeted for degradation with another small protein known as ubiquitin. Rather than tagging with a single ubiquitin, however, proteins that are meant to be degraded are polyubiquitinated. In other words, a single ubiquitin is added, and that's the signal for another ubiquitin to be added, and that's the signal for another ubiquitin to be added. So there are these polyubiquitin chains that are added to proteins that are meant to be permanently degraded in the proteasome. This is the cartoon from your book showing you that we have a target protein with a polyubiquitin chain, and that ubiquitin tagged protein will find its way to the proteasome. The ubiquitin will be removed and recycled to be used on other proteins that will ultimately be degraded. And now the protein that's at the proteasome will feed through that protease-containing core part, cutting each individual amino acid so that those amino acids can be recycled.